Okay, so good morning, everyone. Welcome to the BIO205 final exam review. Congratulations, you made it. We're almost there. Only have one more day left, and then you can take the test and be done with BIO205. I believe Dr. Cotter is either almost done or is done grading the lab stuff, and all those will be imported into your exam, into your grade, so that you can see what you're going to end up with. Just a reminder, you do need a 92% to get an A for this class. But if you get a 90, 91, you still did a very good job. So first off, I'm going to start with some exam logistics. So just a reminder, the exam will be tomorrow morning, Monday, May 9th, starting at 9 a.m. You can either take the exam by coming to DB123 with Dr. Cotter and myself, and you can take the exam there on a laptop. If you don't have a laptop, we'll provide one for you. Or if you have somewhere else you want to take the exam, like the library, your house, wherever, just log in using our normal link for our lecture meetings on Monday at 9 a.m. And then once you're there, we'll make sure everyone gets on Respondus Lockdown browser, and then we'll give you a code that you'll have to enter to start the exam. You're going to have 2 hours to complete the exam. We do require that the cameras be on, but you don't have to have your microphone on. And then if you have any questions or any issues during the test, feel free to just raise your hand using Zoom or in person, and Dr. Cotter and myself can respond to those. Another thing is there are going to be a total of 84 questions, and they're all going to be multiple choice. So no short answer, none of those type of questions. All 84 questions are multiple choice, so you will have your exam grade when you're done taking it. And I'll have the chat open just in case anybody has any questions. I'll just minimize it for now. So what I plan on doing for this morning session, I know it's a Sunday, happy Cinco de Mayo. Try not to take up too much of your time, so I'm sure we all have plans. So I'll quickly go over some of the topics from units 1 through 3, go over some of the key highlights for unit 4, and the rest is up to you for studying, self-study, group study, whatever it might be. Just remember, anything on the study guides for unit 1 through 4 are fair game on the test, and you'll have about 25% of the exam will be unit 4, then unit 3, unit 2, unit 1. So they're each going to be divided in quarters. But the questions will be intermixed throughout the test. You might get a question on unit 1, and then the second question might be something related to unit 3 or unit 4, etc. Okay, so unit 1. So briefly kind of go over Koch's postulates. Again, there's been a long time since we've seen this, so I just want to go over the steps for Koch's postulates. Remember to observe the patient for any signs of illness and to isolate the microbe. Remember, signs can be seen and symptoms are what the patient feels. Step 2, remember to culture and identify the microbe in the lab using different tests. If you recall, we had you remember some of these tests like microscopic, macroscopic, biochemical. Just remembering these tests. We've done most of these, if not all of these, probably most of these in lab already. So you should be familiar with some of these. Again, we're not going to ask you to specifically describe the test. It'll just be maybe a list 2 or 3 different methods you could use to test in a lab. Next, you're going to infect a new host with the same microbe, and you should observe the same signs and symptoms. And then lastly, re-isolate the microbe and identify the same microbe by using the same exact lab tests. Okay, so our favorite part, cell parts, these never go away. So just like before in unit 1, you could be asked questions for different types of microbes and certain things that you could be used to identify them, such as cell walls, budding, scarring, ribosomes, flagella, plasmid. Just know what types of microbes have what types of cell parts. I put some examples here. You can snapshot this or just take a picture of it after my recording's done. Or again, just go back through your unit 1 study guide and you have a good list there. Along with cell parts, also remembering functions, what do the cell walls do, the membranes, what's the plasmid for? We did a lot of stuff with the plasmid. how it can spread antibiotic resistance. Endospores, the 3 layers of the endospore, that's always a popular question on Dr. Cotter's exams. Also like the pili, what the pili are used for, and also what processes they're used for, like we'll talk about here in a little bit. Bacterial shapes and cells. So if you saw a question like this, what I would expect to be on the exam is you might have a couple of questions giving you some pictures, maybe pictures under a microscope, and it might ask you identify the type of bacteria based on the shape. So a coccus, bacillus, staphylo, strepto, etc. Same thing with flagella types. You could be asked to identify a microbe based on the flagella type. And again, one of the biggest things, is to be able to compare and contrast different types of microbes, whether it's prokaryotic, eukaryotic, and viruses, what parts are involved with those. Gram-negative, gram-positive bacteria, we've worked with a lot of those. E. coli as a gram-negative, Staph aureus as a gram-positive, identifying different things like on their cell wall structure, any of the other components that are involved with those 2 bacteria. One is purple, one is pink, that kind of thing. Mold versus yeast, again, they're both fungi, but they each have different characteristics. Should be able to identify each of those. And then different types of viruses, different types of eukaryotic cells like fungus and protozoan as well. But again, just look at your Unit 1 study guide and you should be able to just describe the parts, or if you're given an example, be able to identify the parts and then say this is a eukaryote, a prokaryote, a gram-positive, etc. And then viral replication, we talked about viral replication, specifically HIV and influenza, multiple units. So this is also a very easy question to throw in there because it kind of covers multiple units in one question. Just remember your 6 types of steps for viral replication. You have attachment, entry, uncoding, replication, assembly, and release. And then specifically, because we dealt with HIV and influenza, remember the types of proteins or the viral spikes, such as GP41, GP120, hemagglutinin neuraminidase for influenza, and also describe what it infects, so either the CD4 cells or respiratory epithelial cells, and also how they replicate. HIV using reverse transcriptase, and influenza uses RNA polymerase. That was a quick rundown of Unit 1. So starting on Unit 2, going over environmental conditions, specifically, understand if we're giving a set of parameters. So say, hey, this bacteria has been living in a fridge at -3 degrees Celsius. What kind of bacteria could this be? Same thing with the oxygen, understanding if I give you a picture such as this, where different types of microbes will grow. You can also be asked, hey, if I give you an oxidase test or a catalase test, what do those indications mean if it bubbles versus doesn't bubble, that kind of. And then just more biochemical conditions. I can't say I really I've seen these specific questions on previous Dr. Cotter exams, but she could always throw something in there. It's a very easy 1-point question, giving you a pH value or a salt value and identify the type of microbe. But I'll tell you, temperature and oxygen are very popular questions. Genetics, we did a whole chapter on this one, whole lab on this as well, understanding the 3 different types of horizontal genes transfer between conjugation, transformation, and transduction. Understand conjugation uses that plasmid that we talked about earlier, and it uses that sex pilus, which we also talked about earlier, and how it's able to share its DNA into plasmid and sometimes transfer antibiotic resistance. Transformation, that is when small pieces of a cell are added to another cell and it gets integrated into their DNA. You don't have to know any specifics more than just that portion. And then transduction uses a virus like a bacteriophage to then accidentally steal an old bacteria's DNA and then move it over to a new bacteria's DNA, which gives it antibiotic resistance, such as E. coli. There we go. Antigenic shift versus antigenic shift. Basic takeaways for this: understand that drift is very small. It's a slow time, it's a slow change, and the viral spike protein does not have that much of a difference. But then shifts are major changes in the spike protein and usually happen because of genetic recombination, such as a swine or a pig being used as the middle ground between a bird virus and a human virus. Methods of control. So remember your methods of control from Unit 2. If you attended my Unit 2 exam, I went a little bit more detail into this. I'll share all of the unit reviews I did as well, because those were a little bit more detailed. But in general, being able to look at a problem and say, how are you going to control this infection? And being able to identify, give me a few physical methods that will sterilize, give me a physical method that will disinfect. Same thing for the chemical methods. I broke it down this way because it's a little bit easier for me to answer that question. But you can be given a question, say, give me a chemical method that sterilizes, give me a chemical method that disinfects. Again, it's multiple choice, so you might see a question that says, hey, if it's a chemical sterilant, I put aldehydes, I might put pine sol, ethanol, and UVC, right? So one is a sterilant, but it's physical. The other two are disinfectants, but they're chemical. So being able to identify which are sterilants, remember the O's, aldehydes, halogen, and hydrogen peroxide, and then which are disinfectants. That way makes your multiple choice questions a little easier. And then antibiotics, I would review this. I don't think you'll be asked a lot of questions on antibiotics, maybe one or two. But what probably asked more likely is what kind of antibiotics or what antibiotics target on a cell, especially because it's going to be the multiple choice questions are still going to be organized in a case study method, case study way. So maybe something asking you about, hey, what kind of antibiotics could I possibly give to target a peptidoglycan cell wall, for example? Those are all your cillins, amoxicillin, penicillin, that kind of thing. Just looking over that list, pick a few antibiotics. Again, I don't really expect it to be too antibiotics heavy. And if there's something that you don't have time to study for, maybe study this at the end, just in case you run out of time. Unit 3. So we're going to talk about basically the big takeaways for Unit 3 are the lines of defenses. First line, second line, and third line. First line is basically any of our physical or chemical barriers, such as our skin, respiratory system, the eyes, GI tract, and reproductive system. As you can see here, I broke it down into physical and chemical barriers and organized them by where those barriers are. For the most part, if you look at something like physical barriers, you'll notice, respiratory tract, GI tract, reproductive organs, they mostly have mucus and mucous membranes. So they're very similar. Respiratory tract and reproductive also have cilia, so that's a very easy way to remember it. And respiratory tract, also remember that we have coughing and sneezing as a first line of defense. That's usually forgotten. And for chemical barriers, a little bit more, a little different than some of the other ones, but just going over your chemical barriers and physical barriers. Again, a question could ask you, identify one physical barrier that's used to protect you from a respiratory infection. So then you could list mucous membranes, mucus, cilia, coughing and sneezing. You could also have one of those questions where it groups them together. So you might have mucus and cilia and that kind of thing. Resident microbes. Right now, we've talked about a lot of these resident microbes, Staph aureus, Staphylococcus, E. coli. Just maybe identify one or two in different locations, just in case this question pops up in the exam. Also understanding how your flora protect you, right? How they basically block other pathogens from attaching to you. They compete for nutrients, and they also inhibit other antibiotics, if you will, that can attack other harmful bacteria to keep them away from our body. Second line of defense. I really do expect this to be on the test, especially because this was something that was struggled with on the Unit 3 exam. Understanding that neutrophils and macrophages are involved in every immune response. Doesn't matter which one it comes from, it's always involved in every immune response. Know which chemicals are associated with each of the second line cells. You can see macrophages can do a lot of different things, but some of the other ones like neutrophils and eosinophils, just digestive enzymes, basophils and mast cells, histamine, that's what makes us very itchy. And then understanding what processes are activated by the chemicals. Interleukin 1, interleukin 6 with fever and inflammation. That's a very popular one. Also the membrane attack complex or the MAC attack. And then understanding phagocytosis, anything that's digestive chemicals means it needs to help it eat, which is what triggers the phagocytosis process. But if you're not comfortable with second line defense, I'll guarantee you'll have at least a few of those on this exam. Third line defense, same thing. This is something that people struggled with, especially with the receptors. So in the Unit 3 review, I kind of broke down a strategy for doing these kind of case studies. Always the first question you want to ask yourselves is the attack inside the body, or sorry, inside the cells or outside the cells? Depending on where the attack is going, you'll branch into one of two different methods. So an intracellular response, like cancer, or like a virus, you need to be able to attack the virus from inside of our cells. And so that's always going to use Th1 because you're number one. And it's going to use the CD4 helper T cells. And it's going to communicate using the CD4 plus receptor and the T cell receptor because you're still using T cells. Imagine a CD4 cell, one cell. It has a T cell receptor on one end and a CD4 receptor on the other. So the macrophage can talk to it with the CD4 plus receptor, and then it can go talk to something else, another T cell with a T cell receptor, to then activate the cytotoxic and memory T cells. And then T cells, what they'll do a little bit more in depth, you don't really necessarily need to know this for the test, but it might help you remember, is those, it's called a majohistocompatibility complex, or MHC. So what those are basically just flags. Each of our cells has a flag that tells our body, hey, I'm good, I'm healthy, I'm okay, don't worry about me. It's like a passport. I can come in and out through the TSA just fine. When our cells become infected, they will flip on a different flag, another MHC that basically tells our body, hey, I'm infected, I'm not okay, need to get rid of me. And so the macrophages will screen, look at this guy's passport and be like, oh, you're not good to come in. If anything, we need to get rid of you. And that actually triggers our body to kill that cell for the good of the rest of the body. But understand that just T cells, once they're activated, they start checking all the MHC, I believe it's MHC1. Don't quote me on that one. I have to go back and look at it. It's in our notes though, but understand that it's going to go screen all the MHCs on our cells, and it's looking for that specific infection. And then once it finds it, it'll basically kind of shoot it with perforins and granzymes to cause it to pop. And then once our infected cell pops, all of the viral DNA junk is no longer protected by our cell membranes, and then our macrophages can come in and eat it all up. And then extracellular response, that's anything that's too big to go in our cells. So basically bacteria, fungi, protozoans, those are all considered extracellular attacks or a priority 2. And that means you need the Th2 helper T cells to respond to that. And again, it uses the CD4 plus receptor and the T cell receptor, or the TCR, to communicate those messages throughout the body. The difference between both these responses is that the extracellular response, it's so big, either bacteria or the fungi, it's so big that our cells can't necessarily pop it open, not directly. So what it'll do is it'll activate our plasma B cells and memory B cells. So instead of attacking this microbe, what our plasma cells release are antibodies, and these antibodies are very specific to that bacteria or that protozoan. And we'll start doing is as it finds that bacteria, those antibodies start attaching around that entire bacteria. And what it'll do is it'll block the bacteria from being able to attach to something else. And then once it attaches one bacteria, more antibodies could attach something else, and then they connect those two bacteria together. And then a third bacteria comes in, gets attached together. And it creates this big clump. And remember the antibodies, what they kind of do, they absotomize, they basically flavor the clump of bacteria and antibodies to make our body more likely to want to go and eat and destroy that. So by doing that, you inactivate the bacteria, you kind of concentrate it a little bit, which makes it a bigger target for our macrophages to then come in, find it, and eat it all up. The biggest thing with antibodies, though, is antibodies do not destroy or kill anything. They just do the four things that are listed here. With a glutamate, which is the clumping, they neutralize by blocking the bacteria from interacting with anything else. They opsonize, which is the flavoring of the bacteria to make it more tastier for our macrophages. And then complement fixation is just like another way to trigger complement proteins to basically start breaking apart the bacteria. And then just briefly remember your antibody types. Which is your biggest antibody? Rich IgM, M for massive, if you want to remember it that way. Which antibody goes everywhere? IgG. That can also cross the placenta. Things like IgA can be used for respiratory or mucosal infections. So just look over those to remember which situations trigger which kind of antibodies. I will guarantee that's on the exam. Okay, Unit 4. So Unit 4, I know we didn't really spend a lot of time on this one. Only really about two lectures or so. It is about 25% of the exam, but it also relates to stuff we covered in Units 1 through 3. The first part that's very important for the exam is going to be the question 4, the infection chain. You're going to have one, maybe even two questions on this infection chain. It's going to start from, can you identify the causative agent, which is always a microbe in our class, the reservoir, which is where is that microbe living or hiding or sleeping on. So things like contaminated food, water, dirt, another person could be a reservoir or another living organism. And then the modes of transmission, understanding once that microbe is in a reservoir, how does that microbe jump to something else, another host? Examples again, like vehicle transmission, which is you eating something or drinking it. Vector transmission, so that's using things like mosquitoes, fleas, or ticks. Indirect contact, which is more like the nail, stepping on the nail. And the example of that, people didn't really understand it, was you have the nail, but you have dirt around it. So your foot, even though you step on the nail, it's not the nail that's causing the infection, it's the dirt on the nail, which is why the nail is the indirect contact. The dirt by itself wouldn't get into your body. It kind of needs the nail to puncture your skin to allow the dirt reservoir in your body, which then releases the causative agent in your body. I just put some examples here. By now, all of these should hopefully be filled in. Anything on this chart is fair game for what we spoke to about in class. You can forget Lyme's disease because that was my class. We didn't talk about it in this one. If you don't have this, please reach out to me today so I can at least send you this completed document, and that way you have it to study. I don't see anything in the chat, so I'll keep going. The stages of infectious illness. So how would I ask this question? You might be given something like a graph like this, and you might be given a point on the graph saying, hey, what stage of illness is this? Or you could also be given a description, and then you point out, hey, this is the active part of illness or the decline phase, convalescence phase, etc. So I very vaguely described it here. Inoculation, just like we did in our lab, we took our microbe, we plated it, and it would reproduce. Same thing, the microbe gets in our body. It's got to start probing around, starts reproducing in our body. Initially, our immune system might not even know it's there. And so it'll start reproducing in our body until it becomes big enough to start causing our symptoms, which is where the prodromal phase starts. As we start feeling I don't feel good right now, but you can't really describe why. You might have some light headaches or some pains, but overall you don't have the specific symptoms that say you're infected with this. It's more of the generic symptoms of pain, or maybe you're a little tired, maybe a slight elevation in temperature, but not necessarily a fever, that kind of thing. Active illness, that's when you're hit hard, you're sitting on the bed, you've got the sopa, you've got all your little warm drinks like tea. Dang it. Sorry. But that's just when you're the most sickest. That is when the infection is the strongest. And if it's not dealt with at that point, that's usually when you can start suffering either very aggressive infection that could result in either like loss of limbs or potentially even death. Decline, that's when your immune system finally catches up and is able to deal with the infection, either in the immune system by itself or with the help of antibiotics or some other treatments. And then convalescence is the infection's kind of worn off, and now the body can switch from I'm in an immune response phase to let's switch the body to start repairing the damage that this infection caused. Again, this chart is very important. I've starred these charts because they always pop up on the exams. So being able to identify what the difference between an endotoxin and an exotoxin, you might be asked in a list form like this, or it could be a question saying, hey, this baby was infected with, I don't know, E. coli, and it's got LPS on its cell membrane. What kind of toxin could be released? Something like that. If you see a question like that, just be able to identify between the endotoxins and the exotoxins. Again, if you don't have this, please let me know and I will send you my copy. And then the same types of exotoxins, I'd probably expect definitely one, maybe 2 or 3 questions on this, basically given the type of toxic or the type of toxin and kind of where you might find it or what it could do for you, do to your body, such as like with the blood, the cytotoxins, such as the horny bacteria, neurotoxins and enterotoxins, that kind of thing. So just being able to have a list of these toxins and then understand where they could be found and then what bacteria, what causative agent could potentially be involved in these types of toxins. And I gave you some examples here. And then I believe this is the last point, very briefly going over, just understand the differences between endemic, outbreak, epidemic, and pandemic. You could be given this in a form of a definition where they give you the definition and you have to select one of the 4. Very easy question to throw in an exam to see if you actually understand it. And then virulence factors. This is handout pages 10 through 16, goes on much in detail. But let's understand the 3 factors that we're talking about. There's plenty more virulence factors that are out there, but for this class, we focus on 3. So attachment, such as sex pili or pili and spikes. Again, we talk about virus spikes in units 1, units 3, units 4. So you can see something like this being attached to a question about like an HIV virus or a viral infection in general. Factors to evade the host immune cell, such as the capsule or a glycocalyx coating. Again, that kind of goes back to our unit 1, the cell parts. Different types of enzymes and also antibody receptors, where the microbe has a way to block our antibodies, essentially. And then factors that cause damage, like toxins. Again, we talked about toxins in this chapter. You will have some questions on toxins on this exam. Just checking the chat. Okay, perfect. 6 minutes. Okay, so final tips. Again, anything on the Unit 1 through 4 study guides is fair game. I would also review your case studies or your older exams. That way you can get a breakdown of like what kind of questions could be asked. Questions that were struggled with, that people struggled on, will definitely be asked on the final exam. Again, type the The second line and third line immune defense was definitely something that people struggled with. Chemicals, like how to kill with chemicals and antibiotics, that was also a struggle on Unit 2 exams, so you'll probably see some questions on those. But again, what's nice is that it's multiple choice. So if you don't know the answer, eliminate the ones that you know are not the right answer, and then you give yourself a better chance of at least guessing. And because it's multiple choice, as you go into the test, if you get to a question and you just don't know it, just skip it, go to the next question. Don't stop on that one question and burn your time. Go through the entire test and then come back to the ones you don't know. Something in the test might trigger or help you remember the topic that you're missing. Make sure you have a quiet place with stable internet. If you can't guarantee that, please come to the site. Or the site. Come to the school or get a library so you get that stable internet, that good computer. And that way you don't have to worry about dropping out during the exam. The exam does close at 11 p.m. Things like, or a.m. I think it's 11:05 or something. And then once that exam closes, that's your grade, no matter how far you got. And then the other stuff, the non-academics, get good night's sleep. I know Cinco de Mayo, you probably have some parties planned. I know I do. Just try to, maybe save the celebration for tomorrow and just like drink a few things here or there. So don't come into the exam all tired or hungover or just not feeling well. It's not going to do yourself a favor. You're almost done. Just wait till tomorrow. Eat something, please eat something. And then coffee is your friend, or tea, whatever you need to use to give yourself a little bit of a boost. And then lastly, you got this. You got this. You've done it. You've gone through this entire class. You just did your presentations. You're running a high. Just come in, take this test, and call it good. And you'll be able to go on with the rest of your week. And then lastly, thank you very much for a great semester. You guys were a fantastic class. I appreciate the card and all the fun notes there. Later on, once the school year is over, I'll send my information, just if anyone wants to connect, my social medias, or my cell number, my email, that kind of thing. But good luck on your exam. I wish you the best. I'm going to go through the chat real quick because I've got 3 minutes. Chris, yes, I will send you my Unit 4 study guide, so no worries. And for the success coupon, if you go to the, there should be an assignment link for the success coupon, Araceli. So you should be able to just scan and upload your uncompleted success coupon. If you didn't submit it, Dr. Cotter might have already given you the points. I know if she doesn't see it submitted, she'll sometimes just give the points out for students that she knows haven't used it. She tracks who uses the success coupon throughout the year. So double check that, but if not, there is an assignment link for the success coupon, and you can just send it digitally. Do I have any other questions from anybody? Okay, if no questions, then I'll let you get on with your Sunday. Again, good luck. I will see you all tomorrow bright and early at 9 in the morning. And please message me with any questions that you have or any last minute things that come up. All right, thank you all. I'm going to close the meeting, but have a good one.